health problems associated with it. All right, so those are the two dominant alleles you can have for this hemoglobin protein. And, oops, go back. There we go. Here's what they look like under the microscope versus the genotype. So these are the genotypes you can have, and this is the phenotypes you can have. If you have two of the dominant alleles for the normal protein, you get nice round red blood cells. And if you have two of the dominant alleles for the sickle-shaped version of the protein, you have sickle-shaped blood cells. If you have one of the dominant alleles for normal hemoglobin and one of the dominant alleles for sickle hemoglobin, then about half of your blood cells are normal-shaped and about half of your blood cells are sickle-shaped. And so again, notice this is fitting our description of codominance. There's two dominant alleles, and if you have both dominant alleles, you fully show both traits. You've got some fully round cells and some fully sickle-shaped cells. All right, and incidentally, there's some terminology that describes these various genotypes. If a person has two of the dominant alleles, they're said to have normal blood. If they have, or two of the dominant A alleles, they have normal blood. If they have two of the dominant sickle alleles, they have sickle cell anemia, they have the disease. If they're heterozygous for the two dominant alleles, they have what's called sickle cell trait, where that's where half the cells are round and about half the cells are sickle-shaped. And kind of interestingly, people with sickle cell trait don't have any weakness. They don't suffer from any of the anemia. So it must be that enough of the red blood cells are round and functional for them to be healthy. And so what I want to point out is something here. If you focus your attention on the shapes of the cells in sickle cell anemia, then the normal allele and the sickle cell allele are codominant. And what I mean by that is simply that if you have both of those alleles simultaneously, you show both traits simultaneously, the round cells and the sickle cells. But if instead you focus your attention on the health aspects of the disease, then if you have both alleles, you are fully healthy. You're not halfway healthy or anything. So in that situation, if you focus in on the health, the HbA is dominant and the HbS is what? It's acting recessive if you focus in on health, because here you're getting just the phenotype of the good allele, the health, which gives you healthy blood. You're getting none of the phenotype of the other allele for having anemia, having unhealthy blood that doesn't carry a lot of oxygen. And so it is codominant if you focus on cell shape, but it's just normal dominant versus recessive if you focus in on health. So I guess the message there is you have to be very specific about what part of the phenotype you're focusing in on before you can say it's non-Mendelian genetics or Mendelian genetics.
So the health aspect would be Mendelian.
Right, with a dominant allele and a recessive allele. Now, on Wednesday I was mentioning that anybody from any ethnic group can get any genetic disease, and that's true. But some ethnic groups, there's a higher incidence of certain alleles than in other ethnic groups, so you might find different frequencies of genetic diseases. And this is an example of that. People from African ancestry tend to have a higher incidence of the HbS allele in the population, and so you tend to get more sickle cell trait and sickle cell anemia in African-derived populations. And, actually I got ahead of myself again. I was going to give you a Punnett square. If two people both with sickle cell trait are going to have kids, what's the chance that their kid will have sickle cell anemia? Is what?
50%?
No. 25% is correct. Because since both parents have sickle cell trait, those are their genotypes. And if you fill in the Punnett square, it looks like that. So every time they have kids, each kid has a 25% chance of having sickle cell anemia. Each kid has a 50% chance there and there, 2 out of the 4, of having sickle cell trait, but that's fine because it's still a healthy kid. And a 25% chance of having only the healthy allele, which is also a healthy kid. But as I was saying, in people of African ancestry, there's a lot more of the HBS allele in the population. And there's kind of an interesting story to that, evolutionary-wise. What they found is that the sickle cell trait gives some resistance to malaria, which is why they think it's so high in Africans. This right here shows a map of where malaria is mostly found, where malaria is the most endemic as a disease. And here's where they find the highest instance of the sickle cell allele. And it almost maps exactly where you find the malaria. And so the parasite that causes sickle cell disease is one that gets inside the blood cells. So somehow having some of the sickled blood cells thwarts the parasite. And so in a way, this is an evolutionary solution to a problem of malaria by having more of the HBS allele in the population. If your kids happen to be heterozygous, that's great because they have some malaria resistance and they don't suffer any health. Health effects. But in a way, it's kind of a devil's bargain because for every kid that you do have, then there's going to be a 25% chance that the kid has sickle cell anemia, which is, if untreated, can be a deadly disease. So you're gambling on healthier kids 50% of the time versus unhealthy kids 25% of the time. Sound alrighty? Okay. Well, let's see. Let's talk incomplete dominance now, which is a different type of non-Mendelian genetics. It's different than codominance. In incomplete dominance, one of the dominant allele is not fully dominant. And here's what I mean by that. Getting back to Mendel and his pea plants again for a second. With Mendel, that was a purple flower. And this was a purple flower, heterozygous. Fully purple. And little p, of course, was a white flower. Like that. Even though this had 2 of the dominant allele and this only had 1 of the dominant allele, it doesn't matter. Even if you only had 1, it made enough of the purple pigment to give you fully purple. But what they found is with some dominant alleles, the amount of the dominant phenotype that you get is dependent on how many of the dominant allele that you get. In other words, this gives you a more intense version of the dominant phenotype than this was, or some people say this gives you a weak form of the dominant phenotype. And it's sometimes called a dosage effect. And I think what that means is this. If you take 1 sleeping pill, a small dose, you'll get a little bit sleepy, but if you take 2 sleeping pills, you'll get very, very sleepy. So it's sort of the more you have, the more of the effect you get type thing. And a classic example of that is a type of flower called a snapdragon, where the pigment that's made in these snapdragons is a red-colored pigment. If you have 2 of the dominant alleles, you get lots of enzyme making the red pigment, so you get a very red flower. If you only have 1 of the dominant alleles, you only have a little of that enzyme, and so it makes a sort of weakened form of the red, which looks pink. And if you have none of the dominant alleles, then of course you're going to be white because you're not making any pigment. So notice that that's different from Mendelian genetics. Mendel would have predicted red, red, white, but instead we're getting red, weak red, pink, and white. All right. What percentage of the offspring will be pink? 50% is correct. What percent will be red? 25%. And 25% white. There you go. Fill in the table looks like that. Yes?
So, like, would it be safe to say that Hitler's leading because he was massacring all these people just to keep the blonde hair and blue-eyed people, but he didn't realize that some people had heterozygous and homozygous traits?
Yes, that's a good way of putting it. So he was trying to wipe out people who didn't have, well, I guess, at least for the blue-eyed thing, in theory that might have worked because the blue-eyed is a recessive allele. Little b gives you blue eyes. And so if you killed off everybody who had brown eyes, all you would be left was the little b allele. So you could make an entirely blue-eyed pot. But I think what you're thinking is this: if he was trying to get rid of the blue eyes and only have brown eyes, then he could never do it because some of the brown-eyed people would be like that, and they would still be carrying the blue allele even though you couldn't see it. Yeah, because blue is recessive. Yeah, but I see what you're thinking. Yeah. Here's another example of incomplete dominance in. Oh, there's the flowers. In human beings, there's a protein that causes the hair to have a curly shape. And it's called the W protein, and it's dominant. So if you're big W, this enzyme makes your hair very curly. And if you're little w, you don't have any of this enzyme, so you have straight hair. But if you're heterozygous, you have what's called wavy hair. Wavy. All right, so if these two people have a kid, what kind of hair is the kid going to have? Wavy hair, right. She's big W, he's little w, W. You get the kid, you're going to have a weakened form of the curly phenotype, which is going to be the wavy haired kid. All right, and lastly, another example of incomplete dominance is skin color in human beings. There is an enzyme, we'll call the A enzyme, that makes a brown pigment called melanin. And this is the same pigment that makes your skin dark. It adds brown, or if you have enough of it, black coloration to your skin. And this A enzyme takes a colorless precursor molecule, modifies that molecule, and changes it into the brown pigment. So if you have two of the dominant alleles, you're going to tend to have a lot of melanin made in your skin. If you have only one, you'll have a lesser amount. And if you don't have any of the dominant allele, you're not going to have any of the melanin made in your skin, and you'd have very light skin. Now, so it's incomplete dominance. It's a dosage effect, again, how many of the dominant alleles that you have. But this is not the complete picture, because if it were this simple, there would only be three colors. Everybody would either be dark-skinned or medium-skinned or light-skinned, and that's not reality. If you just look out on the street, we come in a whole range of skin colors from very, very, very light white to very, very, very black. And so I'm saying there's more than three phenotypes out there. And even if you say, well, some of that's because people spend a different amount of time in the sun, even if you disregard sun exposure, if you just look at newborn babies, there's way more than 3 phenotypes for skin color. And so this is an example of something called continuous variation, or a quantitative trait. And it's a trait where there's a huge range of phenotypes. Instead of having just dominant recessive, or dominant and incomplete dominant and recessive phenotypes, you get this whole bell curve from one extreme to another extreme. And usually what this means is that there's many different genes contributing to the trait. Now probably what I should have said here is many different genes contributing through incomplete dominance to the trait. So in other words, there's lots of genes giving the trait, and each gene has a dosage effect where the more of the dominant alleles you have, the more of the trait you get. And so what final level of the trait you have really depends on how many of the dominant alleles you get. And here's, keeping with our example of skin color, a moment ago I said, well, there's this A enzyme that makes melanin, but it's more complicated than that. In real life, there's at least one other gene, which we'll call the B gene, that also makes melanin. So it's a completely separate gene. It comes from a completely different chromosome, but it has